Good morning, students. So we will continue competition number 3.1. Today's topic of discussion is disaccharides with example and its importance. Disaccharide, all of us know, these are the carbohydrates which on hydrolysis gives two same or different types of monosaccharides. These two monosaccharides are joined together by glycosidic linkage. The important examples of disaccharides are sucrose, lactose and maltose. First we will study about sucrose. Another name of sucrose is called as invert sugar. Now this sucrose is obtained from cane sugar and it is used as a sweetening agent in our diet. It is present in sugarcane, beet and fruits. It consists of one molecule of alpha D glucose and one molecule of beta D fructose. So this is the two monosaccharides of sucrose, glucose and fructose. Here the linkage is formed between glucose and fructose, in which the first carbon of glucose combines with second carbon of fructose to form a alpha beta 1,2 glycosidic linkage. So in this case, at first carbon of glucose molecule the functional group is present, and in case of fructose the functional group is present at second carbon atom. So as the linkage is formed between first and second carbon atom of these two monosaccharides, both the functional group involves in the linkage formation. Hence there is no free functional group. Therefore sucrose does not give Benedict test positive and it is a non-reducing sugar. This is the Haworth projection formula for sucrose. So this is about the non-reducing nature of sucrose. I have already explained. Now why it is called as invert sugar? When you take a solution of sucrose in a test tube, then it is dextrorotatory. After addition of acid or enzyme to the solution, if you take again the optical activity of that solution, you will find that the same solution which is previously dextrorotatory now becomes levorotatory. This change in the optical rotation of the sucrose solution after adding acid or enzyme is called as inversion, and hence sucrose is called as invert sugar, and the enzyme which bring this inversion is called as invertase. Now what is the mechanism of inversion? Sucrose is a dextrorotatory and it has optical activity plus 66.5 degree. When we add acid or enzyme in the test tube of sucrose solution, then this sucrose solution is hydrolyzed to form glucose and fructose monosaccharides. These two monosaccharides have independent optical activity. Glucose is a dextrorotatory having optical activity plus 52.5 and fructose is levorotatory having optical activity minus 92 degrees. So if you compare the optical activity of glucose and fructose, the optical activities of fructose is more as compared to glucose. Therefore in a test tube which contain equimolar quantity of glucose and fructose, it shows levorotatory optical activity. Sucrose initially, as I have told you, it gives Benedict test negative because both the functional groups have involved in the linkage formation, but when it is hydrolyzed to glucose and fructose, they become free from each other. The functional group of glucose and functional group of fructose are free and they give Benedict test positive. Now importance of sucrose. In our body, sucrose is digested or hydrolyzed by enzyme called sucrase. It is present in the intestinal epithelial cells. Sucrose is not present in the blood. Therefore, intravenous infusion of sucrose does not digest the sucrose in the blood, and this sucrose will remain in the blood for long period of time until the kidney filters it and excrete it. Due to presence of sucrose in the blood, it increases the osmolarity of the blood, and by the process of osmosis it drags the water from the nearby cells into the blood. Now this property is used to treat a patients of cerebral edema where there is accumulation of fluid in the brain, and by the process of osmosis this fluid is drawn from brain to the intravascular compartment or blood. Another importance of sucrose is, in some rare cases the enzyme sucrase is deficient. It is a inborn deficiency. Such person will not able to synthesize enzyme sucrase. Therefore, if such person eat the sucrose in any form, it is not digested into the epithelial cells of small intestine and hence it is not absorbed. Only monosaccharides are absorbed. Sucrose is a disaccharide, therefore it is not absorbed from intestine, and then sucrose enters into the large intestine. In the large intestine, bacterial flora ferment this sucrose into acids, and this acid then increases the peristalsis movement leading to loose motion, abdominal pain and flatulence. Flatulence means gases formation. Next disaccharide is lactose. It is also called as milk sugar because it is present in milk. It is consisting of one molecule of alpha D glucose and one molecule of beta D galactose, and these two monosaccharides are joined by beta 1,4 glycosidic linkage. So this is the diagrammatic representation of two monosaccharides present in the lactose. Here you are seeing glucose and galactose. The linkage is present between the first carbon of galactose and fourth carbon of glucose. Here the first carbon of galactose is the functional group which has involved in the linkage formation, but the first carbon of glucose is free. This is the diagrammatic representation of Pyranot's formula of lactose. As I have told you, in lactose one aldehyde group of glucose is free, therefore it gives Benedict test positive, and lactose is a reducing sugar. In the small intestine, this lactose is digested by an enzyme called as lactase. Now importance of lactose. A clinical case is seen which is called as lactose intolerance. It is quite common as compared to sucrose intolerance. It is due to deficiency of enzyme lactase. In case of lactose intolerance, lactose is not digested due to deficiency of enzyme. Therefore, it is not absorbed, and this lactose enters into the large intestine where it is fermented into acid by the bacteria present in the large intestine, and this acid then causes loose motion, abdominal pain and flatulence. There are two types of lactose intolerance. One is called as congenital lactose intolerance and another is acquired lactose intolerance. In case of congenital lactose intolerance, the enzyme lactase is deficient since birth. In case of acquired lactose intolerance, the enzyme is present at the time of birth, but its synthesis does not occurs in the body as the person have not drink a milk for long period of time. So this lactase is a inducible enzyme. That means it is synthesized only when the substrate is available. Such person who have not drink a milk for long period of time suddenly drinks a milk, the lactase is absent, therefore he suffers the symptoms of lactose intolerance, but it is a temporary condition. As soon as he drink the milk, the synthesis of lactase in his body starts and he can able to digest the milk after few time or after few days. So this is about a lactose intolerance. Next is maltose. It is also called as malt sugar. It is obtained by partial digestion of starch. It consists of two molecules of alpha D glucose, and the linkage present between these two monosaccharide is called as alpha 1,4 glycosidic linkage. So this is the two molecule of glucose. Again here alpha 1,4 glycosidic linkage is formed between two glucose molecules. So first carbon atom, which is the functional group of one glucose, combines with the fourth carbon atom of another glucose, hence one functional group is free. Therefore it gives Benedict test positive and it is a reducing sugar. This is the Pyranose formula for maltose. So this is about the reducing nature of maltose. It does not have any importance. Only thing is that baby food contains maltose because it is easily digested, as it is a partial digested product of starch. So this is about the disaccharides, their examples and importance. Thank you very much.